everyone, this is Richard from Modern Healthspan. As part of his research on rejuvenation, Professor Goya studies cryonics, the freezing of human tissue such as brain, with the hope of having it restored at some future time. We had the chance to talk with Professor Goya about his insights on cryonics, and as an introduction, I wanted to run through the paper that he and his team had recently published.
Here is the paper, Cryopreservation of a Human Brain and its Experimental Correlate in Rats. You will see that Professor Goya is the main author. Cryonics is still in its infancy now, but several countries have established self-help groups with a mission of cryopreserving human bodies. In this study, the authors reported on the cryopreservation of the brain of a human and a complementary experiment in rats. The human was a 78-year-old woman whose brain was extracted and preserved after death before being shipped to the US for a long-term storage. The paper goes into some detail on the preservation procedure, and Professor Goya talks through it in our interview. At the same time, Dr. Goya and his team performed similar operations on rats, either leaving the brain unfrozen, using a vitrification solution, or using the same protocol as that used on the woman. They then unfroze the brains and assessed the health of the tissues. The patient's brain was successfully preserved and shipped to the US. The paper goes through the details of the checks they performed on the rat brains, comparing the frozen ones using two different techniques with that of unfrozen brains. I will not go through the results in detail as I do not understand the brain physiology well enough, but their conclusions seem to be that the morphology and structure of the brain was well preserved. The understanding that deep freezing a living creature irreversibly terminates life is now being challenged. In our discussion, Professor Goya talks about some examples of resuscitation such as mammalian embryos, tardigrades and nematode worms that can be frozen and then brought back to life, apparently unharmed. So it does seem that it is possible that if a human body or brain is cryopreserved after a natural death, we may at some time have the technology to reanimate it. And as the authors note, even NASA is looking at the technology as a way to preserve people over long periods for spaceflight. For me, this last application is actually the most exciting. Could people be put into suspended animation for long-distance spaceflight?
I hope that you found the video informative. Please do watch out for our interview videos with Dr. Goya on cryonics, where we go into more detail on the processes involved and the science behind them. Please do hit the thumbs up button, subscribe to our channel, and hit the bell button and choose all for any new video release notifications. Thank you so much for your kind support. I wish you all well and will speak to you again soon.